to BioPharma Finder 2.0 review on how to use the new intact workflow to do target calculation for a ADC molecule. So on my screen, what you're going to see is we're in the protein sequence editor, the manager, where we're going to put our protein sequence in, and this is for intact analysis. And the molecule that we're actually going to look at is cadcylan, and you'll see that I have the light chain, 2 copies of the heavy chain, and another copy of the light chain. Now, if you remember, this is extremely important for intact analysis, that you have the proper number of chains to give you the correct molecular weight of your protein. And you can find the molecular weight of the protein, of the target protein, here in this area. You'll see the monoisotopic mass and the average mass. Now you can go through and link together the disulfide bonds, and that can be done by double-clicking on not double-clicking, so you do get static modifications, but right-clicking to create a link of one cysteine to another cysteine, and you link them together. However, in this example, what I've done is I've kind of taken a little bit of a shortcut, and I knew that there were already 16 disulfide bonds in this molecule. And I know that whenever I form 16 disulfide bonds, I lose 2 protons at the formation of a bond. And so I can create a variable modification, just create a new modification. It doesn't have to be a variable mod, but a new modification. So you can see I did that here. I called it DD bond 16, and I'm going to subtract 32.25 for forming 16 disulfide bonds. So I kind of took a shortcut. Instead of going through and linking all the disulfide bonds, I can apply this new modification that I made as a static modification on this cysteine here. So if I double click on it, you'll see that I've added the 16 disulfide bonds. So I'm kind of taking a shortcut there. So, and it's a little bit quicker when you do a lot of these intacts. It helps speed you along. So I have that, so I've taken care of my disulfide bonds. Now for this complex molecule, because we can see the different glycoforms, and then we also have the different drug, so we decided this is a little bit of an easier way to handle the data and interpret the data as well. And so the approach I took here was, in addition to creating this disulfide bond modification, I created the Glycan's modifications as well. So you'll see there's a whole bunch of them in here in purple, and in this case this is G0 and G0F. So on one of the heavy chains there would be a G0, and then on the other heavy chain there would be a G0F. So these are combinations, these are kind of pairs, and these are singlets. So what I've done to simplify things is I also, as well as creating those glycan pairs, is I created a modification for the drug, so DM1. So what I'm doing is I'm adding the drug as a variable modification, and I have it set to 8 max modifications. I'm not using the N-glycan here, but what I am doing is I'm assigning the N in one of the heavy chains, and I'm putting on a glycoform as a static modification. So if I double click on here, you'll see that I assigned one of the static modifications for the glycans, and you'll notice up here my name is that glycan. So I hit OK, and I come up and I change my name, and then I save it. Now what I'm going to do is I'm going to do this for each of the glycoform pairs that I expect to find. So I would just double click, come up, pull down, grab my other pair, hit OK, and then go change my name and hit save. And then what you'll end up with when you actually do this in your list, you'll see you have 6 or 7 different forms of the molecule. And see, you'll see that we have 8 maximum number of modifications, and that's going to be for the drug molecule. And then we have each one of the glycoforms. Now we can select all of these when we actually do our annotation, so it'll be a lot easier to actually do that. So now let's go into the intact workflow, and I'm going to navigate in here. I have to find my raw file. So this is the new intact home page. I hope you like it. And what you'll be able to see is that you can easily navigate to your raw files. And so here you have your raw file. You can name it. So this is going to be very similar to peptide mapping, where you're going to actually name an experiment. Now I can bring in multiple files. Let's see if I actually have 2 files. Here we go. Let's use these so I can demonstrate how you do multiple files. I can assign these to different conditions. I can say run 1, run 2, and let's do run 1, run 2. We're going to do multi-consensus in this example. And then here's where I would actually pick up all of the different forms of the cation molecule with the drug as the variable modification. So I selected all of those, and I'm going to select, we're just going to pick our, let's try our native average over RT for now, and we can always change that once we get into the software. So we're going to do manual processing. So now we're inside of the software. This is the new process and review page for the intact. And under real-time optimization, right here, you have all the parameters that you need. So hopefully you won't have to navigate around in the software to optimize your parameters as you process your data sets. In this example, this is native mode for this molecule, so we have our salt peak back here. I'm just going to click. So when I click here, you'll see that these are the 2 raw files, and then you can see the 2 source spectra. And when I do this first, the other thing you can do as well that's kind of neat in here is you can pop out all your windows and move them around. You can make that a little bigger if you want to see. So I'm looking to say what kind of mass range would I like to use in my deconvolution. So the first, I have to get a source spectrum. Get my source spectrum, then I'm going to deconvolute it. And so I just clicked on one single scan here, and maybe I'm going to click, maybe I'm going to say I'm going to look at 5500 to, I don't know, maybe we can come out to this region right there. So I'm going to change my mass range. That's going to be one of the first parameters I want to change. And this parameter is actually now for the chromatogram and deconvolution, so we only have one mass range parameter now, as opposed to the other software used to have several. Let's set that to 6600. So that'll give us the bulk of this signal. So we're going to do that, and we can just do an average over RT. So I can, you can toggle back and forth. Here's a little toggle button, just so you might see that. You can zoom out, you can zoom in, so I can zoom, or I can double click this. Let's see, I can reset. I can do averaging, so you just toggle back and forth. You can average across over this chromatogram, so you can see it a little bit clearer now. This is where you would turn on your sliding window as well, so you could toggle back and forth between these quite quickly. So you can see how just averaging over a specific region, averaging over a specific region will give you results, and then you can see how that compares to the sliding window. So it's very quick and easy to go back and forth between the two. But we'll go ahead and set some sliding window parameters. So I think I can say I'm going to let my sliding window go from 3.5 to 5.5 over this region. I'm going to leave it set to 1. I believe I'm going to set some offset of 29%. I'm going to set this to 12 ppm, and we'll leave our max gaps to half of a minute. And minimum number of detected intervals for our sliding window will be set to 3. So these are the parameters that are going to help us generate our source spectrum. Now, once we've generated the source spectrum, we have to deconvolute it. And so now let's go to our deconvolution parameters. In this example, it's average mass. So we're going to use where it's unresolved, it's unresolved peaks. So we're going to use the respect algorithm, which will generate an average mass for us. So we're going to, here's your basic respect parameters. Here's the advance, so you can go into the advance if you need to. Often what I like to do is I like to narrow down my model mass range if I know the mass of the molecule. And in this case, I do have a good idea of what the mass will be. I'm going to set as a while I'm dealing with masses, I'm going to set the target mass of my molecule. In this case, I have a pretty good idea. It doesn't have to be exact, but the closer you get, the better the results can be. For native data, I'm actually going to set the minimum adjacent charge states 4 to 4 for the 2 different mass regions in this example. And I know for this specific data, I'm going to look at the charge states between 20 and 30, and I'm going to narrow the mass tolerance down to 12 ppm. I'm going to allow it to use the resolution, which in this case is going to be 49,000, which will be just fine. And I think that's all I'm going to change. Now I can come to my identification tab. So that was our component detection. So that's creating a source spectrum, deconvoluting the source spectrum. Now we're going to identify the masses that we generate. So we selected these 7 or 6 proteins at the protein sequences at the beginning. Now what we want to do, we want to do the DAR calculation, so we need to enable this. So enable antibody to drug ratio. Now the software is going to tell you, wait, you need to assign a variable modification candidate. So in this case, what I wanted to do is I want to use the drug. So I'm going to select the drug. I'm going to have my sequence matching set to 20 ppm, which will be fine. And I do have multiple files here, so something else I want to consider is whether or not when I merge together my results from my 2 experiments, these are the parameters that are involved in that process. So while I'm going to go ahead and get this started. So it's going to recommend some parameters for us. So based on our sliding window, so if I use a 0.1 minute width for my sliding window, it's recommending that I use an 80% offset. So we'll go ahead and run with the recommended. Now you'll notice that it's moving across the sliding, the little windows moving across the first run. This is the source spectrum. Now it's also doing the deconvolution as well. So as it moves across there, it's deconvoluting the data as it's moving. Okay, and we already have our results. That was, wow, super fast. Okay, so again, these windows are the same as peptide mapping. And so you can move things around. You can pull things up. You can move things all over the place. You can kind of set them however you want to. In this example, maybe what I'll do is I'll put this other window over here. These are kind of our ID windows, and these are kind of our spectrum windows. So this is a very quick, fast assessment of this data set. And what you're looking at is here you're seeing the chromatograms. Here you're seeing the convoluted spectrum for both raw files. So we have the top and the bottom file. So in this case, it was 71 and 72. You'll notice that we have the DAR underneath. So the average DAR was 3.28 for that file, and the average DAR for this one was 3.37 for this file. And you'll also notice that all the data, all the deconvoluted spectrum are labeled with the identification. And you'll see that it has the drug load here. So this is a D3. It tells you what the actual protein identification was. So remember how we named the proteins the different glycoforms. That helps us know which glycoform we're actually looking at. And then you can see 3X because there were 3 drugs on this specific mass. So it does help when you're naming these proteins for interpreting complex data. That helps to do that. And then you have your source spectrum underneath, which is very important. So you can go back and look at that one. Now let me explain these 2 tabs over here, because these are quite important. Let's start with the match sequence one. So this is the one that will show you when you click on a component down below, it's going to be linked up to the windows above. And so you're seeing the blue is marking the deconvoluted spectrum, the mass for that specific component. This in the match sequence is showing you here's your mass, here's your intensity, this was the drug load number, and here was the identification. And if you scroll over, you can see the error in the actual identification. And you can always slide all these windows around. And there are filters everywhere. So if you want to filter, if there was more than one thing in here, you could filter. You can also uncheck the identification as well, and you can turn it back on. So if you don't think that's correct, it will remove it from here, it will remove it from here, and you can save it. And it will be gone. So we'll just turn that back on. And down in the table down below, you'll see that you have all your identifications. So the table has a ton of information that you can export out into Excel, and you can filter and so on. So I hope that you'll find the table a little bit easier to use in this version. Now there's this average DAR tab, so if we click on this one, this is where the actual DAR values, the average DARs are actually coming from. And how you use this table, scoot it over a little bit so you can see everything. On the top, you're going to get an experimental average DAR. So this is looking at both of the raw files and pulling together that information. You can see the individual raw files. So that's what you have here, so you can do a comparison. And these will match what you see on the spectrum. And then this table here is actually showing you the information, the raw data that's being used to calculate the average DAR. And when you first process it, the software is going to use all the information. But what's really cool is, let's say that you want to see how the DAR, the average DAR value is per glycoform. Or, and again, these parameters are not optimized, and I'm not seeing all the different drug levels, but what we can do is, again, we can filter. So let's just kind of play around in the table here. And so that just shows me no blanks, and you can see the identifications that I got. And the most abundant glycoform that I have here is G0F, G1F, which we expect. So I'm going to filter this and say contains. So you just click on there. There's a whole bunch of different options. Hit contains. Do G0F, oops, I always make this mistake, G0F underscore G1F, okay? And so we're missing the 0 and the 7. We probably needed to optimize those parameters a little bit better, but this is great. This is just enough for the demo. So you'll see that I have from 2 to 7, which is great. So let me check some of these. Okay, so got those checked. Now, once I check those, I can come up here and I can hit recalculate. So watch what's going to happen to our table and see our values change. So now I'm actually using this information. So these 6 components were used in, so the raw signal, again, the raw signal from these 6 components were used to do the calculation. Okay, so you can see up here that this component wasn't actually found in that other file. So it wasn't found in 2, but it was found in 1. And it wasn't found in this one either. So we weren't able to see 6 and 7 in the other 5. So we might need to go back and adjust our parameters and fine-tune maybe our sliding window parameters, or I think our deconvolution parameters look pretty good. But we could go back and adjust them and see if we could possibly get some better results on those lower level components, which is most likely with the 7, and then of course the 8 and the 0 drug forms of the molecule will be lower in abundance. So this is an example. Now you can also export this out. So one thing I want to mention is that always right click in the software, because you'll see that you can actually export the data out. So you can export this to Excel. You could export this to Excel. You can do component level or all levels. You just checked or not checked. And then to get back, you can simply just clear this. Okay, and then you could just check them all or hit recalculate, and it will take you back to the defaults. And then you have the completed. So what's nice though is, as you see these, and you can see how quickly that did process, you can now adjust the parameters. So you say, okay, well now this looks pretty good. What could I adjust possibly to get down into to being able to see the 6 and the the 8 and the 0 drug load for the molecule? Is there a parameter I could adjust perhaps to make that better? So you can kind of look into your source spectrum. We could, let's see, we can adjust. I think this is a good parameter. This we might want to bring this up a little bit. So we can say, what if we come up a little bit earlier? I think that'll be okay. Oh, one other thing I forgot to show you that is really important. It's kind of neat to this version. Let's do relative abundance. So I showed the abundant one. Okay, when you open this up, you're going to see we were at the component level. Now we're at the raw file level. Now click on component. What you're seeing is you're seeing the individual raw file level information now. So let's pull this guy over here. Now we can see him. Okay, so now we're inspecting this at the raw file level. And you'll notice this is the base peak chromatogram and this is the abundance trace. So you can easily see these now. In the old version of the software, they were on top of each other, which it made zooming a little bit difficult to see. You couldn't really zoom in very well, but now you can. And so you can kind of look at this and just assess the differences in the base peaks and in the abundance traces for the individual files. So again, if you got to this, when you click on the component level, and that's why we put this level information over here to kind of just help you think about where you are. So when you're in the level, you're comparing the base peak of one to the base peak of the other. When you expand out and click on the individual file, you're looking at that individual file data. And then if you want to go one level deeper, you're looking at the individual charge state. So that information's there as well. Okay. So just a little bit of tricks on what's in the software, and you can see you do get the abundance trace only with the sliding window, so it doesn't, it's not part of the other processing. Okay, so let's see if I can zoom in here. Yeah, so we could broaden this. You can always set this a little bit smaller if you want. Is another option you have for your sliding window. Maybe I'll bump this back up a little bit. So we can bump that mass up. Maybe let's bump this mass up a bit. And let's see. Maybe we can set these so these are a little bit smaller. And then we can give it a process. And see. Now we can save a method. And once you start changing your parameters, though, you have to process to save your results. So that's one thing I should have done before we got in here, or before I started changing things. When you go into the method of the manual processing workflow, so if you remember on our homepage, we went into the manual mode. When you're in the manual mode, you have to manually save your results. So you want to make sure you do that before you start changing things if you want to go back and be able to look at your other result file. Okay. So, looks like we got a little bit more results here. You can see that the deconvoluted spectrum looks a little bit more complex. So here's where we would save it. So let's say we really like this one. So we could save our experiment, and now we have a copy of our results with our parameters. So it's important that you remember to do that. You can also save your method. So we can say this was our... Say this is the sliding window method, whatever you want to name it. Alright, so we have that. Now, I wanted to show you one other trick. So if you come in here, now you can, you have your deconvoluted spectrum. Let's say that we really like this result. We can add this deconvoluted spectrum to our library. So we can add it to our library. Now we can name this whatever we want to name it. The default shows it up as the raw file, but you can name it whatever you'd like, and then it will go to the library. Let's say that we wanted to take, now you do have to do this at the raw file level. You can't do it at the component level. You'll notice that it grays out when you do it at the component level, but if you click on it at the raw file level, this will be from the other one. Let's add that to the library. Let's click OK. And now we can come over here to our spectral comparison, and now we could actually compare them. So we can check this one, check that one, and now you can see you have them in a nice mirror plot. So now you can come in and actually see the two different files. I think my window is a little bit big there. You can't see the labeling on the bottom one, and my window is too small in my recording. But it will actually list the raw file names. It will list your Spectra name here. So if you want to name it something different, you can. It shows you which type of algorithm you use to do the deconvolution, and so you're good to go. Okay, well, hopefully this was a nice example of how you can use the new BioPharma Finder to do DAR calculation with an ADC molecule. Thanks.